the light reactions. What happens in the light reaction? There are two photosystems, photosystem 2 and photosystem 1. Everything starts from photosystem 2. When sunlight falls on photosystem 2, the chlorophyll pigments which are present here, they absorb the electrons. Okay. There is a reaction center here, P680. It absorbs at 680 nanometers. A pair of chlorophyll pigments is present here, which receives the electron. When it receives the electron, it gets excited and becomes P680 star. This means the excited state. This P680 delivers its electron to electron carrier molecule, pheophytin. From here, the electrons are transferred to QA, to QB, to plastoquinone, to cytochrome B complex, iron sulfur protein complex, plastocyanin, and finally the electrons are delivered to P700. Now, meanwhile the electrons are being transferred, the protons move out and they keep on accumulating outside the membrane. By the end of this electron transport chain, we realize that there is a lot of H plus accumulated outside the membrane. This H plus accumulated outside creates an electrochemical gradient. Electrochemical gradient means electrical plus chemical gradient. Electrical gradient is formed because of more plus ion outside the membrane and less plus inside, more positive outside, less positive inside. Chemical gradient is because of more hydrogen outside and less hydrogen inside. Now this gradient which is established, like we'll call this as chemiosmosis. The H plus molecule tries to enter back inside. The H plus molecule is outside the membrane. This H plus molecule tries to enter back inside. But because the membrane is semi-permeable, it won't allow entry of the charged molecules. So now this H plus will take help from ATP synthase. ATP synthase is an enzyme which can convert ADP to ATP. It has two subunits. One subunit is a tunnel-like thing, and another subunit are three blades, which when they rotate, ADP converts to ATP. So now the H plus molecule approaches ATP synthase and asks that, can you please allow me entry inside through your tunnel? This ATP synthase says, okay, I will allow you entry inside, but in return, you have to give me your proton motive force.
Yeah.
Now, H plus molecule enters inside this tunnel-like thing of ATP synthase, enters inside like this, and goes back to its original location. Meanwhile, the force which is generated by this H plus, the proton motive force, is used by ATP synthase enzyme to convert ADP to ATP. And that's how ATP is generated.
Yeah.
ATP is generated now. Now, next is about the NADPH. The electrons which were transferred here till plastocyanin, they were transferred to P700, photosystem 1. This is the reaction center of the photosystem 1. It absorbs at 700 nanometers. The pair of chlorophyll pigment which is present in the reaction center, it absorbs the electron coming from plastocyanin. It gets excited, reaches the higher level. The star means it is carrying the electron. Now this P700 delivers the electron to iron sulfur proteins. From here it goes to ferredoxin. From ferredoxin, the electrons are delivered to NADP plus so that NADPH can be formed. And that's how NADPH is formed. And this is the whole light reaction generating ATP and NADPH.